All right, it's time to get to work here this morning. Welcome back. I'm the one who's been out of town, but it's good to be back here and good to have you guys back after that second exam for the Bio 1120 class. Let me just tell you a couple quick things. The average is essentially 78.5% for the second test. My recollection is the average for the first exam was about 72. Congratulations, 6.5% for the class to go up means that things are working pretty well here. I like to think that this means that you guys are taking me at my word when I say look at those older tests. And whatever you've been doing different between the first test and the second test, at least on average for the class, seems as if there are changes in the right direction. Honestly, I don't think the second test was at all easier than the first test. The level of difficulty, as far as I'm concerned, is I'm having a hard time talking this morning. It is at least as difficult, the second test, as the first test was. And I think you might be able to argue it was more difficult. And yet, nonetheless, the class average is up 6.5%. Congratulations, good work. I'll also tell you things that I see when I look at the grade distribution like this is I take heart from the fact that there are 4 of you who got perfect scores. That makes me think the answer key was probably correct, and it makes me think that it was a reasonably fair test overall, because at least a few people were able to agree with me totally about what I thought were the right answers. So, what more is there to say about that second test? Answer key's been posted on Pilot. Maybe the thing that I should say at this point is, there's a final exam for this class as well. It's not too soon for me to start preaching to you that it's going to be very similar to the one that's on the Pilot page for the course. My recollection is the final exam is on Tuesday, December 13th. From 8 till 10 a.m. I don't have any control over that. That's when the registrar has decided, and the registrar decides like 3 years in advance when the final exam for this class will be. So sorry, got to get up a little bit earlier than usual on that day perhaps, but 8 till 10 on Tuesday, December 13th. The final exam is worth more points than the first 2 tests were. The first 2 tests are worth 90 points each. The final exam is going to be worth, I think it's 116 points, okay? And here's the difference. I've had a couple people asking me, is the final exam cumulative? And the short answer to that question is yes, it is. But it's not necessarily as cumulative as you might think and as bad as you might think. Here's the deal. For that final exam, 116 points. That works out to 58 multiple choice questions. Yeah, all right? Did the quick math there. So there'll be 58 multiple choice questions. What I want to do is I'm going to have 45 of the 58 questions be about things that I'm going to start talking with you about today. Okay, so 45 of the 58 questions will be exclusively based on things since the second exam. And then for the remaining, 13 questions, I want to divide those up pretty evenly between what I see as each of the 3 parts of the course. Right, so the stuff that led up to exam 1, the stuff that led up to exam 2, and the stuff that's leading up to the final exam. So 13 divided by 3 is about 4 and some change. So about 50 of the 58 questions are going to be about stuff that I've talked about starting today. Okay? And the remainder will be cumulative sorts of things. What I would suggest you do to get ready for those cumulative questions is, first and foremost, look at the old tests on the Pilot page for the course. That's going to give you a real good guide. But what you might want to do beyond that is just go back to the exams from the first and the second part of the course and take a look at those. Make sure that you're comfortable. I want to encourage you to go back, and if you think you missed some questions, go back and figure out what it is that tripped you up or what it is that you didn't get quite right, and there's a chance that I'll just take questions off of the first exam and the second exam and plant them on the final exam to incentivize you to do that. Okay? So that's how the final exam is going to be cumulative. I hope you don't have to spend a lot of time going back to the first and second part of the course. I hope that you guys, by focusing on the concepts and the underlying sort of principles and the way they get applied, that stuff is going to stick with you. And the cumulative part, I hope, will be very easy. And I hope you can focus most of your attention about what I'll be talking with you about starting today. All right? Any questions or comments about the final exam, the second exam? I've gotten a number of questions by email, and I think I've been responding to those pretty quickly and satisfactorily. If you have questions, you want to talk, stop by, see me before or after class. I'm happy to do that. And again, I think you'll find I'm pretty good at responding to emails. But bottom line, good job, everyone. I think this is a very nice grade distribution, really good average. Looks like we're hitting on all cylinders and doing the right things. All right, so let's turn the page. Stop talking about these nasty exam sorts of things and look at some of the good stuff that we have to talk about here today. I've got some really good material for you today, and it only gets better. The lecture on Thursday is going to be fun. I'm going to start talking with you about some of the experiments that led up to Watson and Crick's insights, and then after that, boy, I'm going to be having a great time. I'll be talking with you about how genes get turned on and turned off. Nothing but happy stuff to talk about there. It's going to be lots of fun for everyone. But we can start today by taking things the next step beyond where it is they were when we finished talking about Mendel a week ago. Right? So a week ago, I talked with you about how Gregor Mendel, this humble Austrian monk, had done some interesting work with pea plants. And he figured, it turns out that was a wonderful model genetic organism for him. Short population times, large numbers of progeny, not very expensive for upkeep, and a number of different traits that he could play around with and he could track. Again, really cool organism for him to work with. And he took things pretty far. He published his results as best as he could at that time. There weren't a lot of prominent scientific journals, but he was publishing and he was telling people about his work. And he gave us, ultimately delivered to us, this idea of the law of segregation and the law of independent assortment. These are sort of Mendelian laws that underpin those classic Mendelian ratios of 3:1 and 9:3 to 3:1 and 1:2 to 1 that come up so often when you're talking about genetic crosses, mating experiments. So that's all Mendel, it's all swell. He did his work in the late 1800s. He died in the early 1900s. At the time of his death, not many people noticed or appreciated what it is that he had been doing. It was shortly after his death, though, that others picked up the baton and continued with his work. So to get ready to talk about those others, and specifically I'll talk with you about Thomas Hunt Morgan, let me tell you what it is that Thomas Hunt Morgan came to understand about Mendel's work. So this is a refresher review based on what it is that I talked with you about a week ago when I was talking with you about Mendel. So again, Mendel does his work with pea plants. He does these mating experiments where he controls the mating. He gets these true breeding parental pea plants, here purple flowering and white flowering. He mates them. He finds that the first filial generation is nothing but purple flowering plants. And that gives Mendel, and then Thomas Hunt Morgan, this idea of dominant versus recessive. The purple trait here, the purple character here is dominant to the white character. We talked about how it is that what was going on at a chromosome level was that there's an allele, a version of the gene for purple flowers inside of each of those purple flowering F1 plants. And there's also a gene, an allele, for the white flowering trait or character. in each of those F1 pea plants. These are heterozygous individuals, but I said this a whole bunch of times, Mendel didn't know anything about DNA. He was vaguely aware of chromosomes, but didn't make the connection between genes and chromosomes, and traits and characters in chromosomes. It was ultimately Thomas Hunt Morgan, as I'll tell you later, that made that connection between what we now understand to be where these alleles are residing and chromosomes. So, again, Thomas Hunt Morgan comes into this knowing about Mendel's work. Mendel had explained in his publications about this 3-to-1 ratio for the F2 generation, where you'd get 3 purple flowering plants for every white flowering plant. And Thomas Hunt Morgan had this sort of appreciation of the fact that because of particulate inheritance and dominance and recessive, that there was also a different sort of genotypic ratio. That's not the word that he used because he didn't know about genes yet, but he has this 1 to 2 to 1 genotypic ratio that corresponds to this 3 to 1 phenotypic ratio. The homozygote recessives account for the white flowering recessive plants, but the mix of homozygous dominance as well as the 2 different kinds of heterozygous individuals account for the 3 purple flowering plants. And I suggested to you that Gregor Mendel really focused mostly on what we would call a monohybrid cross. He's just keeping track of one trait: seed color, flower color, seed shape. But toward the end, he did branch out, took it up a notch, and he started to do dihybrid crosses. And in a dihybrid cross, the simple difference is instead of looking at just one character or one trait, you start looking at 2 at the same time, and you look to see how they move in concert, right? So here's an example of one of Mendel's dihybrid crosses. In this instance, he was tracking both seed color and seed shape. And the pea plants that he had to work with came in either yellow-seeded varieties or green-seeded varieties. And they also came in either round, smooth sort of seeds or wrinkled, shriveled little seeds, right? And so what he did is he did mating experiments between true breeding yellow round pea plants and true breeding green wrinkled pea plants. He'd get an F1 generation. I'll bore you right now. If I haven't already, this will do it. The F1 generation from that pea generation with the true breeding plants, one displaying both the dominant characteristics, the other displaying both the recessive characteristics. Shouldn't surprise you, this is the boring part, that F1 generation are pea plants that are nothing but the dominant trait, the yellow, smooth seeds. But then with those F1 plants, he did F2 crosses, and he gets another mating experiment. He gets an F2 generation, and what does he find? He finds this 9 to 3 to 3 to 1 ratio. Most of the peas, and I mean that literally, most of the F2 plants are showing just the dominant trait, the yellow round seeds that we saw up here at the beginning. Some of them are showing the recessive traits, the wrinkled green seeds, but you also get these that don't look like either of the 2 parents. You get some that are green and smooth. Neither of these grandparents up here were green and smooth. And you also get some yellow wrinkled. Remember, before the wrinkled ones were always green. Now you see some yellow. And you get that 9 to 3 to 3 to 1 ratio. The 3s, the 2 different kinds of 3s, you could call those recombinant. They're different. They've got a recombination of the kinds of traits that we had seen before. None of the pea plants, and I mean that in the pea generation sense, had that particular combination. They look different. There's something strange about them. There's been a reassortment of those genes. We've broken the association between the wrinkled and the color of the seeds of these pea plants. right? And so this is one that gives you that Mendelian ratio that you would expect, 9:3 to 3:1. This is what happens if the gene for seed color and the gene for seed shape are on different chromosomes, on different particles in Mendel's way of thinking, okay? So this is the table that Gregor Mendel has set. These are the things and the insights that Thomas Hunt Morgan has to work with. And Thomas Hunt Morgan comes along, he comes onto the stage, and he wants to take this a step further. All right? So Thomas Hunt Morgan wants to take this a step further. And there's some cool human drama associated with what Thomas Hunt Morgan was doing. So in the early 1900s, Thomas Hunt Morgan reads about Mendel's work, thinks this is really cool. Finally, we've answered these questions that the great philosophers were worried about. Why does like beget like? What's up with inheritance? How does genetic material or information get passed from one generation to the next? Thomas Hunt Morgan realizes Mendel's figured out something really important, and he wants to ride that train. He wants to take that work a step further. And so he takes a faculty position at Columbia University in New York. At the time, an extremely prestigious institution, still pretty prestigious, but at the time, very prestigious. He's all puffed up, proud that he's landed a sweet faculty position at a school that's known for its research. And you guys may not appreciate this yet, but I can tell you as a guy who's gone through the different levels of faculty status, you start as an assistant professor, you work your way up to associate professor, you become a full professor, and then it's all, you know, fat and gravy. It's pretty cushy life as a full professor. But as an assistant professor, it's pretty harsh. You know what the difference between an assistant and an associate professor is? The difference is tenure. Right? Once you've become an associate professor, what they do is they've given you tenure. They say that you're okay, you've got what it takes, you do good work. You can stick around here as long as you like. You can do all sorts of crazy stuff, and we'll still let you stay here and keep your job. When you become a full professor, the big difference there is that nobody even evaluates your work anymore. Right? Full professors evaluate other people's work and decide, oh, if you should get tenure or not, or if you should be a full professor or not. Once you're a full professor, it's just over. Nobody can tell you what to do. For goodness sakes, you're a full professor. You're at the top of the food chain. But again, Thomas Hunt Morgan, like all faculty members, starts at the bottom of the food chain. He starts as a humble assistant professor who lives in fear all the time that he might not get tenure. You know what you got to do to get tenure? You got to make some novel contributions. You got to do some good research, and you got to advance some discipline in an important way so that the full professors can look at you and say, oh, yeah, you're pretty smart, you're doing good work, we'll let you stick around. You get 4 to 5 years to do that, right? If at the end of 5 years you haven't impressed the full professors that you're good stuff, you're out of there. Whatever the equivalent of working at McDonald's was in the early 1900s, that's what Thomas Hunt Morgan had in the back of his mind. This is going to suck if I don't manage to do something really cool. And he's looking at Mendel's work, and he says, boy, Mendel's onto something. I can do something really cool if I just work on what Mendel did and take it to the next level. It's got to work. So he places all his eggs in that basket, and he gets excited, and he says, this is going to be the work that I do. He invests all of his resources in doing this, and he has this clever idea. Right? He starts out and he's thinking, what is it that made things work so well for Mendel? And he realizes Mendel had a great choice of a genetic organism. Large numbers of progeny, not very much upkeep. Number of characters to take a look at. Nobody bats an eye when you do forced mating experiments and incestuous mating experiments. That worked out really well. We've already learned a lot from pea plants. He decides he wants to take it one step further, right? Pea plants, old news, let's figure out something more interesting. He starts thinking, he casts around, and for whatever reason, he settles on a different model organism than Mendel did. right? He wants to do something a little bit better. He's learning the lessons from Mendel. He wants to do something a little bit better. You guys all know already the organism that he chose to work on were fruit flies. Drosophila melanogaster is the genus and species. Excuse me. They are abundant all over the world. There are probably fruit flies in this room right now, though they tend to be fairly discreet, right? We may not notice them. If you have a fruit fly buzzing around you, let me know and we can all look at you and enjoy the presence of your fruit fly. But he decides to work with fruit flies. At a first pass, this is a good idea, right? Small amounts, not a lot in terms of upkeep. You know how hard it is to keep a fruit fly fed and clothed? It's not hard at all. They literally eat trash. They eat banana peels. That's like nirvana for a fruit fly if you give them a little bit of mushed up banana. Even in the 1900s, the early 1900s, bananas were abundant, not a problem. So he's got a good budget to work with here. Fruit flies have really short generation times compared to pea plants. This is an improvement, right? If you could do cool stuff with pea plants that had generation times where you might be able to get 4 or 5 crops a year, how much better would your experiments work out if you could have an organism that gave you a new generation every 2 weeks or so? So in 10 to 14 days, he could have a whole other generation of fruit flies. Their lifespan was short, so this is a big plus for him. Fruit flies, wow, if you guys were impressed with how many baby pea plants you could get, fruit flies just crush the number of progeny issue. You get hundreds of baby fruit flies for every mating experiment between just one male and one female. Wow, this is a home run. And so Thomas Hunt Morgan is coming into this thinking like, man, I nailed it. I got this amazing organism. I've got these neat insights from Mendel. And people aren't all that interested in pea plants, but boy, flies. This is an animal. This is moving in the right direction. It's getting closer to what humans and dogs and cats are. This is going to be cool. He's really jazzed. He's excited about what he's going to be doing with these fruit flies. And he has tons of fruit flies from all over New York City that he can work with. But there's a fly in the ointment. There's a problem that Thomas Hunt Morgan has. You know what the problem was? The problem, Mendel could go down to the farmer's supply shop in his village and he could get like 10 different kinds of pea plants. Short pea plants, tall pea plants, purple flowering, white flowering, green seed, yellow seed, 10 different varieties. There's no farmer supply store equivalent for Thomas Hunt Morgan with fruit flies, right? Every fruit fly that Thomas Hunt Morgan had to work with looked for all the world like every other fruit fly that he had seen. Every fruit fly looked alike. So what's job number 1 for Thomas Hunt Morgan? Job number 1 has to be, he's got this awesome genetic model organism, but there's one problem. He doesn't have any traits that he has different characters for that he can play around with and do mating experiments with. They all look alike. Job number 1 for Thomas Hunt Morgan had to be, and was, find some variants of fruit flies, right? Find one fruit fly that looks different from another so that he can do genetic experiments and see what's responsible, how it is that difference in the appearance got passed from one generation to another. That's got to be his first job. It was his first job. He had a graduate student whose sole job it was to set out traps, fruit fly traps, all over New York City to collect fruit flies in the hopes of eventually finding one fruit fly that looked different from all the others. You know what a fruit fly trap is? You can set these up at home. It's fun, actually. You can do this on your own. A banana peel in a brown paper bag is an awesome fruit fly trap. This is Thomas Hunt Morgan's fruit fly traps. Banana peels, brown paper bags, all over the place. You come, you sneak up on the bag, and you close it, and you look inside, and there are fruit flies all over that banana peel. Works a day, just fine. They set up tens of thousands of fruit fly traps. I don't know what they did with all the bananas, right? But the peels were set up all over New York City. And wouldn't you know, the first month of setting up all those traps, they got like 100,000 fruit flies. Every one looked exactly alike. Crap. Throw them all away, right? That's not very good for us. They go out to the second month, 100,000 some fruit flies. Every one exactly the same. Thomas Hunt Morgan's got to start to be getting a little bit worried right now, right? The third month, everyone the same. The fourth month, everyone the same. The fifth month, the sixth month, the seventh month, everyone the same. There's got to have been a point where Thomas Hunt Morgan is thinking to himself, all right, this isn't working out so well. I've got this, it's called a tenure clock. It's ticking, right? I have like 4 years to do something that's going to impress the full professors at Columbia that's such that they give me tenure, and I'm getting absolutely nothing accomplished because all I'm doing is ridding the city of New York of random fruit flies, right? Can you imagine, and I mean this really seriously, put yourself in Thomas Hunt Morgan's shoes. Can you imagine the sheer joy, the ecstasy, where after 1 year of trying to catch fruit flies, one that looks different from the others, after a full year, he finally finds a single fruit fly that looks different from every other fruit fly he's looked at. He finds a fruit fly. Most fruit flies have pretty red eyes. He finds a fruit fly that pops out. It's got white eyes. Obvious difference, obvious physical difference. He finds a single fruit fly that's got white eyes. Can you imagine the sheer joy? Finally, he can actually start to do some genetic experiments. He can mate this male white-eyed fruit fly with a red-eyed female fruit fly and see what kind of baby fruit flies he gets. He's been waiting for this for a whole year. Cool, huh? Are you with me? Are you feeling his joy? What do you do when you have that kind of joy? You have a party, right? Don't you? You know, this is a cause for celebration. And Thomas Hunt Morgan throws this awesome party to celebrate finally finding a white-eyed fruit fly. And he has it in a special vial so that everybody who comes to his party, which is being held in his lab, he has it in a special vial in his lab so everybody who comes can see, this is my white-eyed fruit fly. Lots of people come. He makes sure all the full professors from Columbia come because he wants to impress them and he wants them to see all the work that went into this. He has a massive party, and as is sort of the norm for academics having parties, there's a lot of champagne involved, right? They're having a good time. One of the full professors cracks open a bottle of champagne, the cork goes flying out, and guess what? It creams the vial that has the white-eyed fruit fly, and the white-eyed fruit fly, as fruit flies do, flew away. Ah! Can you imagine now the anguish? You go from this absolute joy, and then this old fogey of a full professor lets your fruit fly get out, and it flies away. What do you suppose you do at this point in time? The party's over for sure, right? Everybody out. Don't step on any fruit flies, whatever you do, right? Leave. And they set up fruit fly traps like you wouldn't believe in the hopes of being able to recapture that white-eyed fly. And guess what? They got him back like 2 days later. They catch this white-eyed fruit fly, the boy white-eyed fruit fly, and then no more parties. We're getting to work. You in the tube with the female red-eyed flies. We need baby fruit flies fast, right? It's all business, all serious. Let's just start having sex and see how this white-eyed fly trait gets passed from generation to generation. So this is a true story. I'm not making this up. That's how it played out for Thomas Hunt Morgan. And here is a picture of one of the descendants of that white-eyed fruit fly. Again, it's a striking, obvious difference between the red-eye trait, the red-eye character, and the white-eye character. The trait here is the eye color. The characters are red and white. Obvious difference between the two. And Thomas Hunt Morgan, finally, after a year of laying the foundations, is in a position where he can start to say, how is it that the information that's responsible for the white eye color gets transmitted from one generation to the next? A little bit of jargon, nomenclature for you here. If you're looking at a trait and a particular character that most fruit flies have just out in the wild, we got a fancy word for that particular character. That's called wild type. Okay? This is the wild kind of fruit fly eye. This is not. This is something you would probably refer to as a mutant trait, a mutant character. It's different, obviously different from what you see in the wild type individuals. And so he's working here with a mutant fruit fly, and he mates it with a wild type fruit fly, and he's getting to work. Remember, you can't do a genetics experiment. You seriously, literally cannot do a genetic experiment unless there's mating involved. We're talking here about how information gets transmitted from one generation to another. That's what genetics is all about. You got to have that transmission for you to be able to seriously say you're talking about genetics. So he takes his male white-eyed fruit fly. You guys know that symbol, I hope, right? If not, let me introduce you. A little circle with an arrow poking out of it. That's male. A little symbol with a plus sign dangling off of it. That's female. He takes his white-eyed male fruit fly. He mates it with they're being modest here. He actually did about 100 female red-eyed flies because he wasn't taking any chances. But he mates it with a red-eyed female fruit fly, and look what happens. In the F1 generation, right, this is your P generation. In the F1 generation, all the fruit flies have red eyes. Okay? Now, if Thomas Hunt Morgan hadn't appreciated, hadn't understood what Mendel had learned, this might have been another bummer for him. Like, oh no, where did the white eye thing go? But Thomas Hunt Morgan looks at this and he's cool. He says, okay, I think what's going on here is whatever's determining eye color, whatever's determining red eye color must be dominant to whatever's determining white eye color. And I'm going to keep my fingers crossed, and I'm going to hope that in an F2 generation that I'm going to start to see some white-eyed fruit flies again, right? That's what Mendel would have predicted based on his pea plants. If fruit flies are following the same rules, this could happen. So the obvious next thing to do at this juncture, you got a whole bunch of F1 fruit flies. All of them have red eyes. Some are male, some are female. What do? Thomas Hunt Morgan does an F1 cross to get an F2 generation, right? Obvious next step, given what he knew about Mendelian genetics. F1 cross, he takes one of these female flies that have red eyes, he takes one of their brothers, incestuous, right? He takes one of their brothers, he hooks them up in their own private little tube with banana squeeze inside of it and lets them go to town, and he comes back about a week and a half later to see what kind of baby fruit flies he has, right? Pretty quick return on his investment. Here's what he sees for the F2 flies. Big sigh of relief, right? Because look at this. This is a novel observation. Mendel's laws that had so far only been shown to work with pea plants. It looks like Mendel's laws, independent segregation, in particulate inheritance, Mendel's laws apparently are not limited just to plants, not limited just to pea plants. These insights look now like they apply to other things as well, including more complicated things like animals, insects, fruit flies, right? So right out of the gate, this is looking good. Hooray, I've got something. This might work out, all right? And just like Mendel would have predicted, he gets a 3-to-1 ratio. In the F2 plants, this is that classic Mendelian ratio. 3 of the dominants to 1 of the recessives. Sweet, right? Exactly what you would have expected. Thomas Hunt Morgan, appreciating Mendel's work, would have been thinking that, okay, I know the genotype of the white-eyed fly. That's the recessive trait. It's got to be homozygous, right? I'm good with that. I don't know the genotype of the red-eyed flies. It's one of two things. It's either homozygous dominant or heterozygous, where the dominant trait is masking the recessive one. All right? So, 3-to-1, looking good. And then, one of his that graduate student, the same guy who was at the party collecting the fruit flies, that graduate student says to Thomas Hunt Morgan, there's something kind of strange going on here. It's real easy to tell the difference between a male and a female fruit fly. Female fruit flies are a lot bigger than male fruit flies. And again, you guys did this experiment to some extent in lab last week. You got to see some male and female fruit flies. They're pretty easy to tell apart. All of the white-eyed flies were male. Oh, well, what's up with that? Right? 3-to-1, yeah, that's cool. I see what's going on there. But why are all the white-eyed flies male? And this gets Thomas Hunt Morgan to thinking, there's something going on here. There's something strange. It's not just particulate inheritance. This is an inheritance that seems to be somehow associated with the sex of fruit flies. What's up with that? How did that happen? What's going on there? And he does some work, he does some research, he checks out sex determination in fruit flies. About all that he could come up with was this, as people had noticed, cytologists would be the type of people these are, cytologists had noticed that in fruit flies, sex is determined or is associated with a particular chromosome within the nucleus. Female fruit flies had 2 X chromosomes, and male fruit flies always had an X and a Y chromosome. Sex determination in fruit flies turns out to be very much like sex determination within humans. Did you need me to tell you or remind you that human females have 2 X's, human males have an X and a Y? It's the same deal. That's about all that Thomas Hunt Morgan was able to say or know at that time about sex determination in fruit flies. And yet, eye color, this mutant eye color gene seems to be associated with the male fruit flies. And this gets him to sketch out what you see on the screen here. This is a cartoon, but this is Thomas Hunt Morgan's way to explain what's going on with eye color and sex determination in fruit flies. He knows an X and a Y is male, an X and an X is female. He knows that white eye color is recessive to red eye color. And he takes a guess. Well, he knows that believe me, he knows this is a true breeding red-eyed fly, right? Because he can't get a white-eyed fly no matter how many times he gets these red-eyed guys to mate. He knows this is a true breeding fly. He presumes that it's got 2 copies of the wild-type trait, so W plus here. W is short for wild-type. Red eye plus means it is the wild type. It's the dominant version, and they're helping you out here by showing this chromosome in red, right, to help you realize that's going to give rise to red eyes. And he guesses that the male fly has an X chromosome that's got a mutant version of the eye color gene and a Y chromosome. And he's not sure what to do with the Y chromosome. He doesn't know if it's strictly equivalent to the X or not, so he just sort of like, I'm not going to say. I don't know what kind of gene for eye color is on the Y chromosome, but he's thinking on the X chromosome is a gene for eye color. The red ones here that he starts with are true breeding. They have 2 wild-type red eyes genes. The male mutant has at least 1 messed up eye color gene. When these flies make eggs and sperm, the sperm could have either the X chromosome with the messed up eye color gene or they could have a Y chromosome. The eggs, there's no choice. This female can only make one kind of egg. They're all going to have an X, and they're all going to have the red eye color gene associated with them. When you hook up one of these sperm, a sperm with an X and a messed up eye color gene, what do you get? You get a female fly. You get that, right? It's female because it's got two X's, one from the dad and one from the mom. That's why it's female, because remember, female, two X's. It's red-eyed because the red eye gene is dominant to the white eye gene. Okay? So this is a red-eyed female. When this sperm hooks up with this egg, what do you get? You get a male fruit fly. Why? Because it's got an X and it's got a Y, right? And it's red-eyed because on the X is that dominant gene for eye color that determines red eye color. But when these guys, the F1 flies, hook up, start having little baby flies, you get to see something a little different, right? The sperm that this guy could make would either have an X with the red eye gene or a sperm that has the Y in here without an eye color gene. And the eggs are either going to have an X, they have to both have X's from the female. They're either going to have an X that has the red eye color gene or they're going to have an X that has the white eye color gene. If you hook this sperm up with this egg, you get a homozygote dominant 2X'd individual female, right? You hook up this X with, you hook up this sperm with this X to this egg with this X, you get a heterozygote, red-eyed female. When you hook up this sperm with this egg, you get a male, because there's an X and a Y. It's got red eye color, because there's nothing that's voting against it. It couldn't even if it tried, right? Red is dominant to white. But when you hook up this Y-carrying sperm with this X-carrying egg that has the mutant eye color gene on it, you get a male fruit fly with an X and a Y and a mutant eye color gene. In other words, a white-eyed male fruit fly. So Thomas Hunt Morgan lays this all out. He makes the presumption that there's nothing of consequence on the Y chromosome. It's just sort of like a placeholder, because he knew that Y chromosomes were typically much smaller than X chromosomes, and was guessing that there wasn't much going on with the Y. So he just guesses that there's no equivalent gene for eye color on the Y chromosome. And he lays this out, and he publishes his paper. And the main point of his paper was simply this. Thomas Hunt Morgan, for the first time, with this doodling and that one experiment with the white-eyed male fruit fly, for the first time, he establishes a connection between inheritance and genetic material and chromosomes. That was big news. That was headline news. That's a big deal. The realization, this is the first time anybody realizes that it's chromosomes that are carrying genetic information, that it's chromosomes that are responsible for storing information and passing them from one generation to the next. Headline news, Nobel Prize level kind of news, certainly the sort of thing that gets you tenure even at a place like Columbia. So a whole year's worth of work just to catch the fruit fly, and about 2 weeks worth of work to do the mating experiments, okay, a month to write it up as a paper. So in about 13 months, he's secured what he needs for both a Nobel Prize and tenure at Columbia, and the rest is smooth sailing for Thomas Hunt Morgan. Agony for a year trying to find the mutant fruit fly that he would work with, and then easy street for the rest of his life once he hits that home run. This was a big deal. Up till this point in time, chromosomes had been something that cytologists had seen, right? When you're looking at with a microscope, looking at the structure of cells, they could see these chromosomes. No big deal. Don't really know what's up about them. Just who knows what they did. Thomas Hunt Morgan is pointing and he says, you know what they do? These are where heredity resides. This is where genetic information is hanging out. It's on these things called chromosomes. Now, Thomas Hunt Morgan didn't know that anything about DNA. It was literally 50 years later before Watson and Crick figure out that it's DNA that's storing that genetic information, right? He knows nothing about DNA. He knows nothing about genes, but he figures out there's a connection between heredity and these chromosomes. And he can start making predictions. Wherever this X chromosome goes, it brings with it the mutant gene for eye color. He can tie it directly to something he can see at the level of a microscope. And it also happens that it's a chromosome that's associated with determining the sex of these fruit flies. Right? Sort of helped him out that that was also in play. And Thomas Hunt Morgan then says, wow, this is cool. Big deal. I've made an important discovery. I'm going to write this up. He thinks he might get a Nobel Prize. He's right. He gets a Nobel Prize for figuring this out. But then he stops and he thinks, yeah, there's still something that's not quite satisfying. There's still more that he needs to figure out. He knows that there are only 4 different chromosomes that fruit flies have, right? And he knows there's a whole bunch of things that fruit flies must be passing on from one generation to another. They got wings, they got legs, they've got antennae, they've got bodies, they've got eyes. Well, how does that work out, right? If every chromosome determined one of those traits, you need a whole lot more than 4 chromosomes. And so Thomas Hunt Morgan then says, gee, I got to figure out where something other than eye color is determined. Let's figure out what's going on. Maybe some genes or some of these heritable things are associated with the Golgi apparatus. Maybe some are with the mitochondria. Well, we got to piece this all together because there's only 4 chromosomes, and there's a lot more characteristics to go around than what you might have gotten. So here it is, just all together, what Thomas Hunt Morgan did. We had seen it, this slide, then this slide, and then this slide separately. Here's a picture of these chromosomes in their glory. These are actually human X and Y chromosomes. Again, Thomas Hunt Morgan knew the wrap on sex chromosomes were that the X chromosome usually was a honker. It was big compared to the Y chromosome. The Y chromosome's minuscule, hard to pick out from all the other stuff going on inside of a cell. X chromosomes tend to be amongst the largest. And this is actual pictures of both the X and the Y chromosome in humans. And while we're talking about X and Y chromosomes, let me digress a little bit. Sort of a fun story in its own right, but it'll become relevant later on as well. Again, the X chromosome tends to be pretty big. The human X chromosome is huge. It's one of our biggest chromosomes, actually. The Y chromosome is tiny, hard to see. There are very few genes on the human Y chromosome, 4-ish. Okay, very small number. There are thousands of genes on the human X chromosome. Not a lot of action with the Y chromosome, lots of information on the X chromosome. There's an interesting problem that humans and other animals have associated with gene dosage. Okay, to function correctly, we need to have a predictable, a sort of set number of copies of each gene. Our bodies are set up to work with just 1 copy of the information on the X chromosome. Okay, so males, we've got no problem, right? We only have 1 copy of the X chromosome. We got no gene dosage problem, right? Everything's cool, but females have a problem. And their problem is they've got twice as many voices about what should be done with the genes on the X chromosome as they should have. They have 2 times the genes on the X chromosome as what their makeup really needs to hit on all cylinders. right? There's just too much clamor coming from the X chromosome. So you know how females in this room, but mammals in general, solve that problem? It's kind of a simple, cool solution. They shut down one of their two X chromosomes in all of their cells. Again, males don't have this problem. We've got one X, and that's it. We're good. Females have a problem, and they solve that problem of having twice as many X chromosome genes as they need by shutting down one of those two X chromosomes. That shut down X chromosome gets a fancy name. You can see it in a light microscope. It's called a bar body. It's still present inside of all of their cells, but it's a condensed, shriveled up version of the X chromosome. And I'll tell you, the genes on a bar body can't be expressed. They're just too shut down. They're packaged up, they're bundled away. Those genes aren't ever going to be used to make proteins. But what happens is that they shut down randomly one of their two X chromosomes. So they get two. They get one from their mom, one from their dad. Fertilized egg undergoes a round of cell division with two X chromosomes cranking away. So you get two cells, each with two X chromosomes. Those cells undergo a round of replication. 4 cells, each with two X chromosomes cranking away. Those cells undergo a round of replication. 8 cells, 16 cells, 32 cells, 64 cells, right? Round after round of cell division. At 64 cells, you've still got 2 fully functioning X chromosomes, but for humans, and cats, it turns out, it's at that point that one of the 2 X chromosomes gets shut down and relegated to bar body status. Okay, so it's at the 64 cell stage of the embryo that one of those X chromosomes gets packaged up and the other one stays as an X chromosome. And that decision happens independently in each of the 64 cells, randomly in each of the 64 cells, but that decision gets faithfully transmitted then for all of the descendant cells of each of those 64 cells, right? So one of those cells shuts down mom's X chromosome and keeps dad's X chromosome. For the rest of the life of the organism, all the descendants of that cell have a shutdown mom bar body and an active dad X chromosome, all right? Or vice versa in a different cell. And what happens then is that explains stuff like calico cats, like the one that's been looming above me for the last 5 minutes as I've talked with you about this. On the X chromosome, there are genes that determine the coat color of cats, the fur color that these cats will have. There's a gene for orange fur, and there's a gene for black fur. Only female cats can be calico cats. How about that? And all calico cats are heterozygous, meaning for the fur color gene, meaning that they either have the orange, well, they have both the orange and the black gene. But at the 64 cell stage of that cat's embryo, sometimes the orange gene got shut down as part of a bar body. Sometimes the black one got shut down as part of a bar body. And you end up with patches of fur that are either orange or black, depending upon which of those X chromosomes got made into a bar body at the 64 cell stage of the embryo. And you see how that plays out during the rest of the life of the organism. So that's how calico cats work, right? If you ever worried about what was up with calico cats, why they have patches of orange and black fur, voila, there it is. Now, we've got just a little bit of time left to do this particular experiment because it's getting cold out there, guys, right? I think it's supposed to be a freeze like on Friday night, right? But human females, there's a neat experiment you can do here. There's a gene that resides on the X chromosome that determines whether or not you make functioning sweat glands, okay? 95% of the genes are for functioning sweat glands, but there is this unusual mutant gene on the X chromosome that causes you not to be able to make a working sweat gland. And I've got a wife and 2 daughters, so I know women don't sweat, they glow. I got it, right? guys, we sweat like pigs, or if we got the mutant version of the gene, we don't sweat at all. Apparently, I don't have that problem. I'm a pig sweater, right? But here's the fun thing, right? Walk around on a nice warm day when women are outside with their swimsuits on, sunbathing, and, you know, check them out, look and see. Because what you may find is you may encounter a woman that has patches of skin that glow and patches of skin that don't glow. And what you're seeing is the same sort of calico cat phenomenon, but now played out in human sweat glands. Same story, happens all the time. I've given you a command to go out and do that experiment, both male and female alike. Go out there, and if people ask what you're doing, very serious work here. You're looking for bar body manifestations of sweat gland production, something like that. We can dress it up and make it sound fancier if you like when you get in trouble, but that'll work for you. All right? So Thomas Hunt Morgan figures out that because he's stumbled upon a sex-linked gene, he figures out this connection between chromosomes and genetic information. And he takes things then a step further, because he says, okay, I got one trait, I got to find some other traits. And so his graduate student all along has been trying to catch more fruit flies. Wouldn't you know it, like within a couple of weeks of getting his white-eyed fly, they catch a bunch of other kinds of mutant fruit flies. What's the deal? Why couldn't he have had one of those, like in the first month, right? But he finds a bunch of other fruit flies eventually. He helps things along, too, by the way. He irradiates some of his fruit flies to try to induce mutations in them. Worked pretty well. But before you know it, he's got a couple of different kinds of fruit flies to work with. He's got fruit flies with white eyes and red eyes. He's got fruit flies with short, stubby little vestigial wings and normal kind of wings. He's got fruit flies that have gray bodies as opposed to mutant black bodies, right? So he's getting a little collection, a menagerie of mutant kind of fruit flies. And he starts to look to see what he gets with dihybrid crosses, just in the same progression that Mendel had done. And one of his first, I think it is the first dihybrid cross that Thomas Hunt Morgan does, is illuminating. It illustrates something, another huge leap forward in the field of genetics that Mendel couldn't deliver. He studies a pair of genes, one that determines body color, one that determines wing size/shape. And he finds out that these two traits are coded for by separate things, but they're connected and yet not connected. He finds out they reside on the same chromosome. They're determined by different genes. Again, he doesn't use the word gene because he's not quite sure what's going on, but different particles. But the particles are connected, and the particles reside on chromosomes. They're different. There's a gene that determines body color and a gene that determines wing shape. And when he would do these kinds of mating experiments between a heterozygote and a homozygote recessive, again, in fruit flies, the plus means it's the dominant trait. When he would cross a heterozygote with a homozygote recessive, most of the offspring were either what you would expect or what you would expect, a heterozygote or a homozygote recessive. But that's the rub. It was most. And this gave Thomas Hunt Morgan another cause to scratch his head and stop and think. So let's see what actually played out for Thomas Hunt Morgan. These are his actual results. He takes a wild-type heterozygous female fruit fly. The female part is incidental here. These are not sex-linked traits. But he takes a heterozygous individual. I'm sorry, this is a homozygous individual. 2 copies of the wild-type gene for body color, gray body, and 2 copies of the wild-type gene for wing shape. These are normal fruit fly wings. And he mates this pretty little female fruit fly with this handsome male fruit fly that has some issues. All right? he's got two mutations. He's got the black body color, which is recessive to the gray body color, but he's true breeding, homozygous. And he also has these not very attractive wings, right? These vestigial wings. That's another recessive trait for wing colors. He mates this fly with this fly, and what do you expect? What kind of baby fruit flies are we going to get? They're all going to look like mom, right? Because they're going to be heterozygous. They're going to have one good copy for the gray colored gene for body. They're going to have one good copy of the gene for wing shape. So let's keep our fingers crossed. And oh yeah, thank goodness that's just what we find. We've got a heterozygous F1 set of individuals. This has been a dihybrid cross. And now he mates females with dad, again, incestuous. You can get away with this in fruit flies. People frown on that sort of thing in human genetics, but nobody seems to mind when you do this for fruit flies. And what do you get when you do that? Well, let's do the Punnett square sort of thing. What kind of sperm can this guy make? He's a homozygote, right? So every sperm cell has to come with a recessive body color gene and a recessive wing color gene. That's the only kind of sperm this guy can make. So his sperm are over here. What kind of eggs can this female make? Well, she can make eggs that contain the wild type body color and wing shape. She can make eggs that have the mutant body color and wing shape. I suppose it's possible that she could make eggs that have the wild type body color and the mutant wing shape, and maybe some eggs that have the mutant body color and the wild type wing shape. Okay? If you match up this sperm with each of these four different kinds of eggs, this is the kind of baby fruit flies that you would get. Okay? You can skip ahead and look and see that the genotypes match up with those phenotypes. Thomas Hunt Morgan does this dihybrid cross. He sets up the test cross, just like we have up there. And like a good scientist, he's hypothesizing what results he's going to get. Okay? Mendel, for all of the dihybrid crosses that he ever published, got a 9:3 to 3:1 ratio. Always. Every time. And so that would be, well, this isn't quite that same sort of scenario because this is a heterozygote and this is a homozygote. But let's play this out. If the gene for body color and wing shape were on different chromosomes, then each of these kinds of eggs would be just as likely to be made. Okay? You would get a 1:1 to 1:1 ratio of babies if body color and wing shape were on separate chromosomes. The alternative is that they're on the same chromosome. Thomas Hunt Morgan says there are only 4 chromosomes. There's going to have to be a bunch of times the genes are all on the same chromosome. Maybe what's happening here is that wherever the black body color gene goes, the gray body color, the wild type body color goes, the normal wing shape gene must follow because they're on the same, they sit on the same chromosome. The same thing is pulling one where the other is. And if that's the case, if they're on the same chromosome, the only kind of eggs you'll make are this kind and this kind. You can't make this kind and this kind because that's not how they started out. This has them moving independently. If they're on the same chromosome, where one goes, where one wild type gene goes, the other wild type gene must follow, or where the mutant gene goes, the other mutant gene must follow. With me? So Thomas Hunt Morgan comes in, he sets up this experiment, and he's expecting one of 2 different things. Either all the fruit flies are going to look like these two in a 1:1 ratio, or he's going to get an equal mix of this, 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 and this. Cool? See how he's thinking? See where we're at with this particular kind of experiment? If they're on one chromosome, Thomas Hunt Morgan expects you'll get one outcome. If they're on two separate chromosomes, you'll get a different outcome. Easy to discern between the two. I tell you what, if you see something else, we don't know what's going on, right? What's he see? Here are his numbers, right? If they're on different chromosomes, you want 1 to 1 to 1 to 1, right? If they're on the same chromosome, you want just this kind and this kind, right? And none of this kind or this kind. What do you see? These are his actual numbers. He got 9 for just one mating experiment. Again, these are prolific organisms. The technical term is fecund. They have lots of progeny. You get a couple thousand baby fruit flies in this F2 cross. A whole lot of them look like this. A whole lot of them look like this, right? But wait a minute here. Some of them look like this, and some of them look like this. This set of results is different from either this prediction or this prediction. This is not 1 to 1. That's 1 to 1, but that's not 1 to 1, right? This is 1 to 1 to 0.2 to 0.2, right? That's not 1 to 1 to 1 to 1 to 1. I think I might have had one too many ones in there, sorry. And this is not 1 to 1 because there's 1 to 1 to 0.2 to 0.2. Oh, crap. What's going on? Thomas Hunt Morgan thought he had a handle on all this. He's figuring out the chromosomes are involved. He's making these predictions. You're either going to get one type of ratio or you're going to get the other type of ratio. They're either on the same chromosome or they're not on the same chromosome. And he gets this intermediate result. Thomas Hunt Morgan is learning as he's going through, as he's maturing and figuring these things out. And he says, this is an opportunity. This is an unexpected result. I can make another cool discovery. And you know what his cool discovery is here? He decides the only way to explain these results is if the gene for body color and the gene for wing shape are, in fact, on the same chromosome, but once in a while, that chromosome gets broken and the information gets swapped with the equivalent information on another chromosome. In a word, recombination is occurring. Recombination of chromosomes is occurring. And Thomas Hunt Morgan is also, he's a smart guy. He realizes from the previous work from cytologists about sex chromosomes that there may be something going on, so he taps some cytology expertise, and the cytologists tell him, yeah, you know, something like that may actually happen. During this unusual step in cell division, when organisms are making gametes, they go through this thing called prophase 1 of meiosis. And when they're in prophase 1 of meiosis, sometimes we see chromosomes, equivalent chromosomes, overlapping with each other in a way that looks like they may actually be swapping information. We call that a chiasma. And it turns out that there's a bunch of proteins that are involved, and eventually people are going to call that a synaptonemal complex. You guys know what's happening here. This is prophase 1. This is recombination taking place like it had played out a week and a half ago during the course of the lecture. And you know what determines the amount of recombination? It's the physical distance between the two genes on the chromosome. The further apart they are, the more likely you'll have a recombination event. And the larger the number of individuals, the larger the fraction of individuals that look like this. The closer together, the smaller the number of these individuals, the less recombination is occurring. We can call these kinds of fruit flies here recombinant individuals because they look different than either of the two grandparents, right? There's been some recombination of the genes on the chromosomes here. These guys are different. Those guys are recombinant. And you can determine the frequency of recombination really simply at this point. You add up the number of recombinant individuals, and you divide that by the total number of baby fruit flies in that F2 generation and multiply by 100%. That gives you a recombination frequency. I think the math gets worked out for you right here. This slide just shows you the same thing, but now broken down to chromosomes. If you want to track this by seeing where the recombination is occurring, and the swapped chromosomes are shown for you here. But here's the math. There are 391 recombinant individuals, 2,300 total offspring, times 100% equals 17%. There is a 17% recombination frequency between the gene for body color and the gene for wing shape. They're on the same chromosome, but they're far enough apart that 17% of the time a recombination event will take place between them as gametes are being made during prophase 1 of meiosis. Okay? So that's how you figure out a recombination frequency. If this is a big number, it means the genes are further apart. If it's a smaller number, it means they're closer together. In this scenario, you can't ever get a recombination frequency greater than 50%. At 50%, it maxes out because that's what you get when genes are on separate chromosomes. That's complete independent assortment. All right, so you can't ever get a recombination frequency greater than 50%. That just means that the genes are on separate chromosomes. But you can get anywhere from 0% recombination, which means the genes are smack dab on top of each other, maybe a single gene, and we got a word for that, right? That's called pleiotropic, a gene that affects two different characteristics, right? So if the genes are right on top of each other, there is 0% recombination frequency, but you can get all the way up to 50%. That percent, I've been alluding to this poor, hardworking graduate student of Thomas Hunt Morgan's all along here today. His name is Sturdivant. That's his last name. He went on to have a fairly prominent career as a geneticist as well. Sturdivant, as a tribute to his mentor, Thomas Hunt Morgan, suggested, and the name has stuck, that a 1% recombination frequency between genes should correspond. We should put a name on that instead of calling it a recombination frequency or a 1% recombination frequency. Sturdivant said that that should be a centimorgan, right? So a 1% recombination frequency is often referred to as a centimorgan. Curiously, a centimorgan, this is something of trivia for you. It'll become more important maybe later on. A centimorgan, or a 1% recombination frequency, corresponds to about 1 million nucleotides at the level of the DNA of a chromosome. So you have about a 1% chance of recombination for every 1 million nucleotides that separate two genes along the length of a chromosome. So here's the chromosome version of the body color, wing shape story. This is the whole story. Here it is in pieces. I'm just going to fly through this because I think we're good. And check out what you can do with this kind of insight. We know that the gene for body color and the gene for wing shape now are on the same chromosome. We know that there is a certain distance separating them. We can call it 17% recombination frequency. We can call it 17 centimorgans. But we can do the same sort of thing, do other dihybrid crosses with other traits. Here's another gene. This one determines a different kind of eye color for fruit flies. It's called cinnabar. They look cinnamon colored as opposed to the normal red or the obvious white, kind of in between. So cinnabar is a different eye color. And dihybrid crosses with fruit flies that where you're focusing on the body color and this other kind of eye color shows there's a 9% recombination frequency. There's a 9% recombination frequency between this kind of eye color and this kind of wing shape. And you can end up starting to get a roadmap along the length of the chromosome. Not only do you know what genes are on the chromosome, but you have a feel for their relative spacing and the order in which they appear across the length of a chromosome by doing this kind of recombination experiment. Bunch of dihybrid crosses gives you that sort of insight. It seems every week, at least, probably more frequently than that. There's a news story somewhere about somebody has discovered a gene for something or other, alcoholism, pedophilia, diabetes, colon cancer, right? You name it. There's all sorts of things now that have been associated with genes. When people say they've discovered a gene, you know what that means in English? It means they've mapped a gene that's responsible for a particular trait to a chromosome, and they know where that gene is relative to other genes along the length of that chromosome. And you know how they do that? They do that with recombination mapping, like we are talking about here today. Okay? As something as an aside, human geneticists are doing genetics with their hands tied behind their back, right? With fruit flies, it's easy to set up these mating experiments. Lots of incest going on, right? Makes it nice and easy. With humans, human geneticists can't do that, obviously, I hope, right? What they have to do is they have to go out into the world and find people who have chosen to have children of their own accord that also happen to have these traits that they can map and keep track of. So they have to go out and find families, pedigrees, where they can do these analyses and look back in time and say, gee, if I could have had you mate with you, this would have been cool. It gives me some insights. So anyway, that's how genes get mapped. And you can, again, spell out the genes as they appear across the length of a chromosome. Here's a whole bunch of them, right? This is apparently the same genes we had talked about before. Here's cinnabar, black body, and vestigial wings. You can have short little aristae over here as opposed to long ones. You can have brown eyes as opposed to red eyes. All of these genes reside on the same chromosome. These two are 17 map units apart. These two are 9 map units apart, or centimorgans apart. This one and this one are right at the edge of what you might be able to tell with a dihybrid cross. They're at 48.5 centimorgans apart. This one and this one, you know what their recombination frequency is? Again, it maxes out at 50. But by mapping this one to this one, you can extend the map and move along. You can't have a 104% recombination frequency. That doesn't make any sense. You can't have 104% of the individuals. Seriously, right? You can't have more recombinant individuals than the total number of progeny. Right? That's illogical, right? But you can determine that this is 104 map units away by doing this sort of mapping process. All right. Couple last things. Let's talk about chromosomal alterations. There are a couple of words to talk with you about here. This is a picture that shows you how sometimes during meiosis things don't work out the way that they should. You can get gametes made that have an unusual or an incorrect number of chromosomes. Here is one of the outcomes of that sort of thing. This is what you call a karyotype. It's a picture of all of an individual's chromosomes. Okay? So you can get a snapshot of all of their chromosomes, and cytogeneticists are paid big bucks to look at those chromosomes and to figure out what's going on with individuals that have certain combinations of chromosomes. What you see here is an instance of a type of chromosomal abnormality with, it's called trisomy. You have 3 copies of chromosome 21 in this individual. It's a fairly common situation to find. Individuals that have 3 copies of chromosome 21, we say, have something called Down syndrome. And this is an individual with Down syndrome. All right, so here we have, this is called aneuploidy. There's a problem with the number of chromosomes. It's not the normal number of chromosomes. It's not divisible by 2. You've got here 1 chromosome that in all of this individual's cells, there are 3 copies. As I was talking with you before about bar bodies and gene dosage, that's why trisomy of 21 is a problem. You're getting too much chatter from chromosome 21 genes, and that gives rise to the phenotype that we associate with Down syndrome. As something of a bit of interesting trivia to you, did you know that by far the most common trisomy is trisomy for chromosome 21? It is. Chromosome I think it's 17 is another one. I'm drawing a blank on what the name of that is, but it's another fairly common one to have 3 copies of chromosome 17. But all the rest, it's pretty unusual to have trisomy of. You know why? There's a clinical term for people who are trisomic for chromosome 1. It's becoming a running joke with us here, right? We call those people dead. They don't survive as embryos. Those are people who don't get born. We don't see them. We only see trisomic individuals that are not that bad off in terms of gene dosage, at least in the scale of things, right? It could have been a lot worse. So trisomy is a kind of aneuploidy. There are other things that cytogeneticists can see. They can see it when they're looking at chromosomes that a region has been deleted. They can see that within a chromosome, sometimes genes get duplicated. They can see that sometimes that a stretch of chromosome can be inverted. And they can see that from one chromosome to another, some information might be swapped. These are all things that human cytogeneticists look for. And because they've looked at so many different karyotypes for the last 100 years, pretty much any small change in the structure of a chromosome, they can tell you is going to give rise to this set of problems. The suite of symptoms you expect for Down syndrome, but there are others. An inversion might, for instance, cause you to have a child that is prone to bedwetting, be colorblind, and have atrial heart valve defects, right? So that when a parent presents with a child and they do these types of analyses, they can say, oh, and by the way, monitor their heart because there's a chance you're going to have this problem in the future. So these are things that you can do with the insights that we have about cytogenetics. And I tell you what, let's stop there today. I'll just say a few words at the beginning of lecture on Thursday about imprinting. And we'll call it a day. And in class on Thursday, I'll start talking with you about the next steps that got us to the point where Watson and Crick said, hey, DNA is where it's at. All right, see you guys Thursday.